So our speaker today is Daniel Kraushaar. Daniel got his undergraduate degree at the University of Aberdeen before coming to the United States and getting his Ph.D. from the University of Georgia. Then he went to the NIH to do a postdoc for 5 years before he finally joined Baylor. So he was initially the technical director of the Genomic and RNA profiling for the GARP core, which is what he's talking about today. And then he was promoted to a core director in 2020. And in 2021, he also got an appointment as an assistant professor in the MCB, as well as the new Department of Education, Innovation, and Technology. So today, Daniel is going to tell us about transcriptomic and epigenomic profiling and next-gen sequencing, as everyone has access to in the GARP core. Daniel, go ahead.
Yeah, thanks, Steve. So great to be here, and just briefly, what I have lined up is essentially give everyone an overview to the technologies we offer here at the core, and you'll notice there's going to be a recurring theme in terms of there's more than one avenue to address a certain question or scientific problem. So hopefully you can use this to educate yourself and see what options you have and maybe make the right selection. And before I delve into the deep technology, I want to give a higher level overview of who we are and what we do as hope I can move forward. Okay, there you go. So this is our staff lineup. As Steve mentioned me as the director. If you've worked with us, you probably met Emily, our lab manager. She navigates many things in the lab, including sample drop-off and others. And then joining myself in the admin is Q, the QFem. She also does a lot of R&D work. And then we have junior as well as more senior technicians among our staff. We're on the third floor, spread out across 3 labs, and the offices are in 333E. You can find sample prep guidelines, pricing, a lot of useful info on the website. To submit your actual requests or orders, you have to go through iLabs, and down at the bottom here is our email address. Feel free to reach out. This will reach either myself, Emily, or Q. Actually, it reaches all 3 of us. All right, so our primary function is providing you with service. There's a few other roles that might not be entirely obvious to everyone. Generally, we try and be abreast of the evolving technologies that are out there. We're not necessarily developing them, but we're very interested in being the first ones to test them. We want to validate them. That goes for instrumentation or chemistries and kits. With respect to education, we provide consultations, training on instruments to some extent, workshops, seminars. We can help with data interpretation as well. We are a dedicated shared resource for the Cancer Center, also the Digestive Disease Center, and the Center for Precision Environmental Health. So if you're a member of any of these centers, you can get certain subsidized pricing that you want to take advantage of. And then we also get involved in manuscript preparation as far as methods go that we were involved in, technical aspects that we were involved in, letters of support as well. So that's what we do. We operate primarily under a full service mode, meaning you as the researcher, you come to us, you drop off your samples, and you walk away with results. There's obviously a whole lot going on in between, usually consultation, as well as various QC milestones in between. And we typically generate sequencing libraries, as well as we also operate the sequencers. There's some exceptions to that. There is some instruments that you can become trained on and then use on your own. Our Sonicator, and then also some our mid-range or small-scale sequencer, the NextSeq 500. These are the three technologies that we offer, and the instrumentation that goes along with it. There's a nanostring encounter, which is kind of the odd kid here, because it's NGS independent, it's hybridization based, allows for a targeted approach to gene expression, but the output is direct quantitative information. And then two NGS technologies. One is a short read technologies, the Illumina Selecta platform. This is a sampling method, and your end result or your output is short reads containing sequence information. And then more recently, we added the Nanopore platform. This is a long read sequencing platform. So in essence, the output is similar to Illumina, but sequence information, but longer reads.